So today we are going to discuss some more questions regarding this industrial biotechnology, especially focusing on the lectures that you have studied in week 2. Okay, now coming to the first question. This is the question someone asked me last session, that how much glucose is required for producing 200 gram ethanol. Someone please switch off your mic. So first we require to write a balanced chemical equation regarding the same. Like glucose is producing ethanol and CO2. So we must write a equation which is balanced, like all the you all know about the balanced chemical equation, okay? So we must know that 1 mole of glucose is forming 2 moles of ethanol. So from moles you can calculate weight, and from weight here also you can calculate the weight. Then we can know how much, because they are asking in this way, that 200 gram of ethanol require for how much glucose. Okay, so suppose 1 mole of glucose, the molecular weight of this is 180. Similarly for ethanol it is 2 into, because there are 2 moles of ethanol. So we'll consider here its molecular weight is 46. So this is 92. So 92 gram of ethanol is required to produce 180 gram of glucose. Then how much? This is simply a mathematics, that 180 upon 92 into 200. So when you solve this, it will come around 391 gram. So it means that 200 gram of ethanol is forming 391 gram of glucose. Okay. Yeah, this is the answer of this question. So if you feel my speed is slow or fast, please let me know. Coming to the first question of today's session. They have given the 3 figures. Okay, let first read the question. Match the figure with the correct option. In first figure, they all 3 are representing the bioreactor, and different conditions are given there. Suppose in the first figure, this is a bioreactor and nothing is added from the outside and nothing is taken out from the outside. It means like initially you have taken all the things and you have put in the bioreactor. That's it. Then in the second figure, you have seen that S and X means the substrate and other things are required from the outside. Then in the third figure, it is given that substrate and biomass is given from the outside, and it is also removing in a continuous reactor, means the reactor is running and the product and substrate we are removing at a same time. So all of you have studied the 3 types of bioreactor. So which one is which? First one, in which nothing is added from the outside, this is batch reactor. Okay, and in which only the substrate part is added in a running reactor, so that is fed batch reactor, and in which the substrate is added and product is removing at a same time, that is of continuous type of reactor. Yeah, A, B, C. So this is the right answer for your question. Everyone please try to join by 6:05, because otherwise I have to admit in between. Then coming to the second question. For the wastewater treatment, activated sludge process is mostly used. The mode of operation of the process is. So this is also a very simple question. They are asking that whether that operation is batch, fed batch, CSTR, and CSTR with recycling. It means when activated sludge process is used, Then what we have done, mostly the product, the substrate, the substrate is added and the product and the biomass is removal at the same time. And even after the removal, that recycling of this same compound is occurring, because we have to treat in the wastewater treatment, we have to remove all the things we don't require that product or want to collect that product. That's why in this CSTR is used with resell recycling. So answer to this question is D. Okay, coming to the next question. Yeah, this is, what is the degree of reduction for Saccharomyces cerevisiae with the elemental composition of this? This degree of reduction is a really important topic for your syllabus, and if some of you want to give gate biotechnology, this type of questions mostly they ask. So what is the degree of reduction? Suppose this is the formula of any substrate you are considering, or any, let's say biomass or product, we in all the same way we consider that the composition of that compound is in the form of this, that CW. W is mainly representing how much they are. So CW matlab carbon, hydrogen, oxygen, nitrogen, these four mainly come used for the composition of that component. So degree of reduction is mainly found using, mainly represented by gamma, and it is found as the this 4 into W. Why we have taken this 4? This 4 is mainly represent that carbon. For carbon we have take +4. Then for hydrogen it is considered +1. I know all those things you have studied in the lecture, so I'm only helping you in doing the questions and solving them easily. For oxygen we mainly considered -2, and for nitrogen we consider mainly three sources, like if it is taken from NH3 or ammonia, then we consider it -3, and if it is taken from N2, then it is 0, and if it is taken, if the source of nitrogen is HNO3, then it is considered as +5. Here it is -3. Okay, so in this way we mainly found the degree of reduction, and this is divided by W, because we mainly see this degree of reduction for a per gram and per element of this carbon. So that's why we are dividing here with W. So now let's find the degree of reduction for this given compound. That carbon, it is 4. So let's say gamma equal to 4. Then for hydrogen it is 1 into 1.64. Then for oxygen we consider it is -2 into 0.52. How much elemental composition is there, from that will help you. And minus for nitrogen will, if there is not mentioned about the nitrogen source, then by default we consider nitrogen source ammonia. Okay, so we'll take as -3 into 0.16. So when you solve this, it will come around 4.12. Yeah, so answer to this question is B. Even you can read more about degree of reduction, and you can try to find degree of reduction for the other examples. Like this is written here about the degree of reduction, that it is defined as the number of equivalent of available electron in that quantity of material containing 1 gram carbon atom. So we have to focus this, that it is considered for 1 gram carbon atom. And you also have to learn, you can learn or you can find out this, that degree of reduction for CO2, H2 and NH3 is 0. This can this thing you can use directly in some questions or very simple type of questions. Okay, everyone understood this degree of reduction and how we calculate this, because this is really important for solving some other questions also. So if you have any doubt, please ask. Okay, coming to next question. Yeah. The material for of construction of photobioreactor is preferably. So in this question they are talking about photobioreactor. So all of you have studied about photobioreactor. So anyone can tell me what it is or why they are called as photobioreactor? Okay, let's discuss. As a name is suggesting, in this we are using the photo means light we are using. So here light source can be anything. It can be sun or it can be taken from like the inside light. So for taking the light, suppose this is your bioreactor, and if it is made up of, suppose it is made up of stainless steel, then can light enter in this? Preferably not. And if some light even goes, it will go in a very less amount, although it will not allow to pass the light. So that's why we need a material for the photobioreactor that will allow the light or that is transparent. So for photobioreactor, what is the main important thing is transparency. This is the main quality that we need for making the photobioreactor. Okay. So now let's see the options. What are these? First one is stainless steel. Steel will not allow the light to pass. Then borosilicate glass. So glass bottles we can use, as we use glass bottles similarly that will work. Then mild steel, no, it will also not allow the light. Then none of the above. So from these borosilicate glass we can use for the formation of this photobioreactor. Yeah, these are the examples like which we are using in lab scale. Even these kind of bioreactor are also used at a industrial scale for making biofuel, some pharmaceutical components. So you can see that it is mainly made up of glass so that it can pass the light. Even it can also, we can also use the plastic. If it is transparent, then we can also use the plastic. So answer to this question is B. Coming to the next question. Everyone please join by 6:05. Now let's read this question. Which of the following equation is best suited to represent the oxygen demand for a biochemical reaction? So here they are asking about the oxygen demand. And this is really important for many of your numericals and other part. What is the oxygen demand? Knowing that, it is really important to know. So this is the chemical reaction, or you can say balanced chemical reaction, in which a substrate is converted to product and some, sorry, product and some biomass is produced, and other, that CO2 and H2 that are produced, and in this is the nitrogen source and oxygen that is required. So you can see we are finding this oxygen demand, that is B we have to find. So how we have find that? Gamma of substrate. What is this gamma? Gamma is degree of reduction. Okay. Minus, there we mainly consider about the biomass and the product. So how much biomass is produced? That is YC, that representing the stoichiometry coefficient of this biomass, into this gamma of B. Here B is representing the biomass. This small b is considered for biomass, minus, because they are forming, that's why we have taken them as negative. For the product also, this yp, sorry, because we are talking about the product. Then what is the degree of reduction for product? By 4. This 4 is coming from, because for oxygen we have considered degree of reduction as -2, and for, because two elements are there, so this is 4. That's why this 4 is coming. So this is the oxygen demand for any biochemical reaction. You can use this formula in any chemical reaction in which biomass is produced, and even both biomass and product are used. So for this question, correct answer is B equal to gamma S minus YC into gamma B minus YP into gamma P by 4. Yeah, so option A is correct for this question. So this is the next question. For which of the following metabolic end products the degree of reduction is 0? So this is very simple question. Even I told you just 2 questions back that it is H2O, CO2, both H2O, CO2 and none of the above. Even you can find also, but from here you can know that both H2O and CO2 have degree of reduction 0, and along with this NH3 is also having degree of reduction is 0. Even other components also have degree of reduction 0, but these three are very frequently used, so you can know it directly. Coming to the next question. Sorry. Yeah. Our compound contains 62.42% of carbon, 4.64% of hydrogen, 27.82% of oxygen, and 5.12% of nitrogen. So now let's solve this. Yeah. Normally, how we find this empirical formula? This we have also done in class 11, 12th. So you must know that the given percent, it is equal to given percent upon molecular weight. It will give you the molar number of moles. Sorry. Then you can write the formula. So let first start with carbon. But you, okay, we'll discuss it later. It is like 62.42 upon how much carbon molecular weight? 12. So solve this and let me know how much it will come. For hydrogen, it will be 4.64 by molecular weight of hydrogen is 1. So this is 4.64. Similarly, we'll find for oxygen and nitrogen. So these three you will calculate. Please calculate someone how much carbon moles are there. You simply have to divide the given percent with the molecular weight. For oxygen, like it is 27.82 by 16. For nitrogen, it is 5.12 upon 14. Someone please answer.
For carbon 5.2016.
Okay, 5.20. For oxygen and nitrogen, this is very simple calculation.
Oxygen 1.73.
Sorry.
Oxygen 1.73.
Okay, 1.73. Nice. Then nitrogen,
0.36.
fine. So if we consider this will make carbon 5.2, nitrogen 4.64, oxygen 1.73, and nitrogen 0.36. But this is not the answer, or this is not the option given. So we have to read full question, that they are asking what is the empirical formula of the compound expressed in terms of one carbon atom. Okay, this is the main focus of this question, that they are asking about one carbon atom. So we have to, this carbon are 5.2. So according to this, these are there. Sorry, I've written something wrong. Yeah, it is hydrogen. So you have to divide everyone with 5.2. Like we have to consider carbon as 1, no? So carbon, for hydrogen it is 4.64 divided by 5.2. Then similarly oxygen 1.73 divided by 5.2. And for nitrogen also it is 0.36 divided by 5.2. So this will give you the correct answer. Okay. So the correct answer is A. When you solve this, it will come this. So if you have any doubt, yeah, you can ask. Okay, coming to the next question. This is also a very simple question. Which of the following statement are true for a plug flow reactor at a steady state? You all have studied about the plug flow reactor. So what happened in that? I'll give you a brief. That concentration gradient is there, like here there is less concentration than here, it will increase. So concentration is not uniform in this. Okay, let's read the options. First one is concentration of the reactant fluid varies along the path of flow in the reactor. So this is correct. That's why we are using this plug flow reactor, because this is the problem occurring in the continuous and stirred reactors, that they are not coming out at the same time, and the concentration problem is there. That's why we are started using this PFR. So this is correct. Then the rate of accumulation of reactant at a steady state is 0. That is also correct. And there is no back mixing in the reactor. This is also correct for this PFR. So answer to this question is all of the above. So this is the next question. The below schematic represent which reactor? So what is happening in this, that A and B are flowing through this reactor, and this is written here catalytic ceramic membrane. So it means this, suppose this is a reactor and the membranes are like this. They have shown only two membrane, but it is not like that there are only two membrane, one up and one down, and on the whole surface the membranes are bound on the surface. So what happened, then C is forming, and what is A and C are coming out, and this B is mainly diffuse out through these membranes. So this figure is representing the membrane reactor. So answer to this question is C. Now let coming to this question. The conversion of S for a first order liquid phase reaction S2P, that substrate to product, in a CSTR is 60%. If another CSTR of the same volume is connected in series, this is important here, and this is also that same volume. So it means apart from this, other factors will remain same. Then the overall conversion, how much conversion is there? So suppose this is, you all have to read the question very slowly and carefully. Like you have to focus every word, what they are asking and what they are trying to say. Because when you understand the question, only then you are able to solve the question rightly. So let's study this question again. The conversion of S for a first order liquid phase reaction S2P in a CSTR is 60%. If another CSTR of the same volume is connected in series, then the overall conversion will be. So let's consider that this is a CSTR and substrate is given and 60% conversion is occurring here. Then one more reactor is connected in series. What do you mean by that, connected in series? It means that how much product is forming, it will again used as a substrate here, and finally here product is forming. So it means this reaction is occurring twice before forming the main product. From going substrate to product, it is going through two reactors. Here also 60% conversion, because all the conditions of this given bioreactor is same. So suppose here first we have S, then 60% conversion occur. So how much it will go from first to second? It will be 0.4 of S, because 60% is converted, so 40% remaining there. Then this 40% or 0.4 of S will form product. So how much conversion is occurring here? Here we also consider it as 60%. So 60% of 0.4 is converting. It means 0.24 is consumed. So how much it is remaining here? It is 0.16. So what is happening here? From S to P, suppose this is 1, so it means only 1.6 is remaining. So how much conversion is there? So 1 minus 0.16, 0.84, or you can say it as 84% of conversion is occurring, because this is left out after the two reactors. So how much it is converted? It is 84%. So everyone understood this question? You can also get such type of question when they have changed the conditions. So you also have to consider that. Now coming to the next. So answer is B. Next question. Okay, this question is for you. Initially you all have to try this. I'll help you in solving this question, because this type of questions are really important. So you all must know. Every one of you have pen and paper with you. Okay, let's start. Okay, first read the question and I'll let you know how you have to do this. But you have to solve this question by yourself. Let's read this question. Under aerobic condition with glycerol, that is C38H8O3 as a carbon source and ammonia as a nitrogen source, a microorganism with the empirical formula this is produced. So what it is? This is biomass that is produced. Then the biomass contain 10% ash for 1 gram of glycerol. Sorry, the biomass contain 10% ash. So this biomass is containing 10% of ash. How much it is weight? Out of which 10% is ash. And for 1 gram of glycerol, 0.42 gram of biomass is produced. So it means it is 1 gram of substrate is producing 0.42 gram of biomass. No other major metabolite are formed during the process. It means there is no other product is forming here. Then what is the oxygen requirement of the system in terms of ash-free biomass? So what we have to do, first major thing they have given that this biomass is containing 10 percentage. So when you found the molecular weight of this, or when you found the weight of this, then you will consider only 90% of this molecular weight, because 10% is not used. This ash means this is a waste or other component that is not considered with the biomass product or biomass amount. So this is the one thing they have given, and then other thing is given that 1 gram of substrate is forming 0.42 gram. So it will help you know how much moles of this biomass is producing. So what you have to do, you can directly use the oxygen requirement formula, that is that we discussed in the previous question, that is gamma of S minus C into gamma of, that is B, biomass, minus here product, but upon for this term is 0 because no other major metabolite is producing here. Then you have to find these things, that gamma of substrate. So what is substrate here? It is glycerol. So first you have to find degree of reduction for this glycerol. Then you have to find this YC. YC is mainly the stoichiometric coefficient for this biomass. Then how you found this? You can use this equation or this information that 1 gram of substrate is forming 0.42 gram. From here you can find this. Then degree of reduction of biomass. This biomass formula is given, you can directly find the degree of reduction. Simply put it here and you will get the answer to this question. So let's start this. First try to find this degree of reduction of this glycerol. So you guys do this and let me know how much it will come. Anyone please answer. Find degree of reduction of this glycerol. Okay, any one of you solving this or not? Let me know at least. Okay, someone answered 1.16. What is this 1.16? This degree of reduction for this. Okay, I'll also solve this. Okay, I'll show you. First, when we let start from here. Just a minute. When 1 gram of this substrate is converted into 0.42 gram of B, or you are asking the last, okay. First, find out the molecular weight of this. Molecular of this will come around 92. Okay. So when 1 gram of substrate is forming 0.42 gram of biomass, then how much this 92 gram is forming? 0.42 upon, sorry, into 92. It will give you the weight of this how much it is forming. And for finding this YC, molecular weight of this 0.4 into 92 into multiplied by, sorry, divided by this molecular weight of this biomass. So how much molecular weight is there? Okay, let me add one more slide. That 1 gram of substrate is forming 0.42 gram. So first we found the molecular weight for this biomass. This will come around 26.63. It will not come directly, it will come when you consider with 10% ash. Because what is the molecular weight of that? You can find with formula, it is of 100%. But how much weight is there in the biomass? It is 90% only. So when you find out for the 90%, it will come out this 26.63 gram of this is present. So now you can find that Y of C, that 0.4 that is producing into 92 molecular weight of the substrate by 3, because this is for the 3 carbon, because 3 carbon are present here, and the molecular weight of this is 26.63. Yeah, will, because we are finding the mole, so it will come in denominator, 26.63. This will help you in finding this stoichiometric coefficient of this biomass that is producing. So when you solve this, it will come around 0.46. So this is the value of YC. What is the formula that we need? That is degree of reduction of substrate minus YC into degree of gamma of B, that is biomass, by 4. So from this one thing we know, and that, sorry, YS and YB we can find directly from that formula. It will come around, I am telling you the answer, but try to solve this in a step by step manner. This will come around 4.415 and it will come around 4.7. So when you finally solve for this A, or you can say B, because the equation in which you have studied, just a minute, yeah, this is very important equation, you all must know about this and how to solve this. how you are finding this YC, YP, and the one major thing in this equation, it is balancing. Although sir has explained in a very detail in the lectures, so if anyone has not seen that lectures, please go through that, because this equation is really important. This equation will help you in solving all type of questions relating to the biomass production of the other substrate, and even finding the other problems. So here that oxygen is represented by B, and this is A, that is the ammonia source, and this is substrate. In substrate we mainly consider this first one carbon, so that's why this is YC. So when, if it has more than, like suppose 5 carbon, then when we find this, and the gamma will divide by 5, because what is gamma? Gamma is mainly considered for the 1 gram of carbon atom. Okay, so please everyone remember this equation. From this equation only we are finding this B. That formula of B is coming from this equation, that is gamma of S, degree of reduction of substrate, minus Y of C, this is stoichiometric coefficient of C, into gamma of B. B here is representing the biomass, and that gamma is degree of reduction for the biomass. Similarly, if product is forming or not, you can see, or it is mentioned in the question. So it is here is Yp into gamma of P by 4. This 4 I have already explained how it 4 is coming here. So we are only putting the values in this equation. Yeah, here we are solving this. So YC, it is YC, how much is YC is 0.46, then gamma of B is coming 4.15, and gamma of S it is coming 4.7. So when you put all the things in this equation, it will come around 0.709 gram. So correct answer to this question is B. So everyone get this question, how we are solving this type of problems,
Yes, ma'am.
because this is really important. Okay, any other doubt in any other question? You can ask me, because this is the last question for today's session. Yeah. Or if anyone want to give any feedback for this assignment session, then please. Nitin, so now you get the question how you have to solve this and get the right answer. Try to solve it again, you will get the correct answer. And if you have any problem, you can ask me again. Stoichiometric problem. Okay, this one. You are asking from the solved question, or another new question you are asking? Atindra Karan. Atindra Karar, you have asked this na? Stoichiometric problem. Which question you are asking? Solved one. Okay. Can you mention the question number? Period. Yeah, this lecture is also recorded, so it will be uploaded on YouTube. You can see it there, and I will upload one or two more reference from where you can practice the questions. Atindra, can you mention the question number which one you are asking? Question number 7. Okay. This one. The doubt and big to assign. Okay, how we are solving this? It is given a compound contains 62.42% of, sorry, 62.42% of carbon, 4.64% of hydrogen, 27.82% of oxygen, and 5.12% of nitrogen. All the substrate, product, and biomass, whatever we are considering that is forming or we have considered as a substrate, all these we will consider that are formed of carbon, hydrogen, nitrogen, and oxygen. All these four are making that compound. So when they have given the percent, it means they are giving that 62%. Suppose if it is 100 gram, then it is 62.42 gram of carbon is present. This means that 62.42 gram is present in 100 gram. So, but 62.42. For carbon we know, but we have to find the molecular weight. Sorry, molecular weight means number of moles. Because they are asking the empirical formula, and when we write the formula for any component like for glucose C6H12O6, so we'll consider that this much number of moles are present, number of molecules are present in that component or in that substrate. So this 62.42% will divide with 12, the molecular weight of a single carbon. So it will give you the value 5.20. Similarly, you will find for hydrogen, oxygen, and nitrogen. So for hydrogen, it will come around 4.64. For oxygen, because molecular weight of oxygen is 16, so it will come around 1.63. For nitrogen, 14 is the molecular weight, and it will come around 0.36. So if we put all these value that we got this formula, that this is the component, this is the formula for this compound that they are asking, that carbon 5.2, hydrogen 4.64, oxygen 1.73, and nitrogen is 0.36. But what they have mentioned further, that we have to express the empirical formula in terms of one carbon atom, means carbon should be 1 here. So when carbon is 1, then all these values will change. This 5.2 we need to this 5.2 value we need to change it as 1. So how can you how can you change it? When you divide 5.2 with 5.2, this is simply like if 5.2 carbon is same, then it will be 1. So we'll divide this one, sorry, divide this with 5.2. But when you divide carbon, number of carbon, so all these things will change the formula. So for balancing that number, we also divide all these things with the 5.2. So when you solve this carbon will come as 1, hydrogen is come when 4.64 divided by 5.2, so it will come around 0.89. Then how much oxygen is there? Similarly, 1.73 when divided by 5.2, it will come around 0.34. And for nitrogen, 0.36 when divided by 5.2, it will come around 0.07. So this is the empirical formula that they have asked for one carbon atom. Okay. Okay, someone asked, ma'am, Okay, Aishwarya, you have asked about the week 2 assignment 2 question. Okay, because you have to submit this assignment. So first try to solve this by yourself, and when you are not able to do, I'll help you. Channeling effect. Try to read about all these reactors. You can refer two books for this. First one is Doran. Maybe all of you have known this. That first book is by Pauline M. Doran of Bioprocess Engineering. You can refer this book. One more book you can refer for Kargar and Shuri for Bioprocess Engineering. These two books you can refer for this course. In this you can get a detailed for all the things that I have discussed, even apart from this, all the things that is in your syllabus you can find in these books. That channel link or name, uh, we will provide you. You will get a mail through NPTL for the same, or it is simply Learn Industrial Biotechnology. Anyone has any doubt? Even you can ask any doubt or other question in discussion forum. So if you have any doubt regarding the assignment or any other question, you can also discuss it there. I will discuss in the next session.